Welcome back to our lecture dedicated to fundamentals of nucleic acids. So, as we have learned before, there is only one type of DNA in our cells, and DNA is the main carrier of the hereditary information. In contrast, there are three main types of RNA that can be found in human cells. We have the messenger RNA, short mRNA. We have the ribosomal rRNA, and there is also tRNA. Those are the three main types of RNA. However, as we will learn in a few minutes' time, there are also few additional types of RNA. However, they are not of large concerns for you at this stage. So, mRNA is the first type of RNA we need to have a look at. It is generated during the process of transcription. So mRNA contains all of the information on a given gene that are encoded on the DNA, and this information gets transcribed from DNA to RNA. But we will have a dedicated lecture on transcription and replication next week. As I mentioned before, what you need to know for now is that it is transcribed from the DNA within the nucleus. And the mRNA is furnished with genetic code, message, as a series of codon, where each triplet, every three bases, represent one codon. So this sequence, GCU, will produce a different amino acid than this codon, which is ACG. So it's a sequence. Its sequence is complementary to the DNA, and the mRNA carries the genetic information from the nucleus through the cytoplasm. Importantly, this is only the case in eukaryotic cells that do have a nucleus, and that's something you will learn in microbiology as well. Actually, bacteria don't have a nucleus. This is one of the main differences between eukaryotic and prokaryotic cells. The second type of important RNAs in our cells are tRNAs, or transfer ribonucleic acids. This tRNA is a type of RNA molecule that helps to decode mRNA sequence into a fully functional protein. On the right hand side you can see a structure of one of those tRNAs. Please keep in mind that similar to DNA and mRNA, also tRNAs have a clear polarity with a 3' end here and a 5' end here. Those tRNAs have also an additional very important sequence called anticodon. They bind with the anticodon to the respective codon on the mRNA. Please keep in mind that this binding is dictated by the sequence of the bases here. So tRNAs can only bind to the complementary triplet on the mRNA. They also carry one specific amino acid at the 3' end, and this is quite obvious here. We have again our 5' end, 3' end, the anticodon which binds to the codon on the mRNA, and here we see an amino acid attached to this very tRNA. In this case, that's leucine. The last type of RNA, that is the most common one of the three most common ones we need to have a look at, are rRNA. And rRNA are ribosomal RNAs that form a part of the ribosome. rRNAs are only one component of the ribosomes, because ribosomes are composed of ribosomal RNA on one hand, and on the other hand they do have a protein portion as well, and they are crucial for the process of translation, for the conversion of the information on the mRNA into proteins. On an important note here, and we will have a closer look at the ribosomes in our cell biology lecture series, bacterial ribosomes are target of many antibiotics because they do slightly differ from human ribosomes, making them quite specific. So the question here is, how many polynucleotide strands are found in a tRNA molecule? Is it 1, 2, 3, 4? Is it maybe 5? Let's try your knowledge. The answer is 1. A tRNA molecule is basically one polynucleotide strand composed of different bases. So how do those RNAs play together? They actually, all of them, play an essential role during protein biosynthesis. They are all needed to translate genetic information on the DNA into proteins. Again, once the information on the DNA is transcribed on the mRNA, mRNA migrates out of the nucleus and binds to ribosomes that are present in the cytoplasm of the cell. This results in binding of tRNAs that carry amino acids, and a large number of tRNAs binds sequentially to the ribosomes and to the mRNA, forming thereby a growing polypeptide chain, which is then forming our proteins. Good, so as I mentioned before, there are three most common types of RNA in our cells, but there are also quite interesting small RNAs present in our cells. They are not that commonly referred to, and you might not have heard about those guys before at your school. Those guys are very small non-coding RNAs. They are usually between 12 and 22 nucleotides in length, and they are called microRNAs or short interfering RNAs, short as siRNA and miRNAs. So what is the role of those non-coding RNAs? Obviously, they do not contribute to protein biosynthesis, but they play an important role in regulation of gene expression because they are able to block the translation of mRNAs into proteins, or they can induce cleavage of the mRNA, a breakdown of the genetic information. Typically, miRNAs have incomplete base pairing to a target sequence on mRNA, so we might have a match of only few bases. In contrast, as siRNA typically base pair perfectly and induce mRNA cleavage only in a single specific target. So although those two types of RNAs are not necessarily that crucial for this very lecture, we are gonna be talking about those two later on as well, so please keep in mind that those guys exist. However, they are the less common types of RNA. Why are those RNAs so important for you guys as future pharmacists? Because they are actually already used in the clinics. There are multiple clinical trials going on, and the whole sector of pharmaceutical industry is trying to explore this quite heavily at the moment, because we can design artificial microRNAs that are able to target, for example, mRNAs that are produced specifically by tumor cells. We could use them to downregulate the level of expression of certain genes in certain cells, and we can use them as therapeutics against, for example, viral infections. With this outlook, I would like to end up our third session within this lecture, and I will see you in our lecture on replication transcription, which is going to take place next week. Thank you very much for your attention, and please do remember, if you have any questions, use the tablet on Blackboard and I'll be in touch to address those. Thank you very much. Bye.